So let's start with the material. We are going to do the transformation and oncogenesis, and what you are going to see that if you have taken cell biology, you will be super excited with that because you will see that the lot of things will now make sense. And if you are concurrently taking cell biology as well, uh, usually the cancer biology is in your fourth module, and you will see overlapping material. And that's some for many people, that is very exciting to see that how these two things related to virology as over. And overall, I guess that probably the story of the transformation and oncogenesis out of all the 17 chapters that we are going to discuss, probably the most complete and fascinating story in that sense. So really, you are just going to see that we are going to go into the detail of some of the pathways to some extent, and but overall, you just need to appreciate the big picture of the virology, that how the knowledge of transformation and oncogenesis all came together, and how virology paved the path of it, and very, very interestingly. Along with the cell biology. So we're going to hear a great story. That's basically what it is. So let's start. So so the first thing that what you can see that this is almost a repeat if you have taken cell biology. And this is how we actually develop a cell culture. We are constantly talking about the cell culture, and usually how we do this, you can take a hamster or a mouse embryo or something like that. You just chop it up, and then you trypsinize it. So the trypsin is a protease that will separate the cell adhesion, and you take these single cells and then you plate them in a petri dish with the proper media, serum, everything. And what you're going to see that they are going to grow in the petri plates, and time to time when they are growing, you need to passage them, means that you have to change the petri. You take some of the cells and put it in a new petri plate, let them grow, and so on. So this is a typical way that normal cells grow. And what you are going to see that initially the cells will attain a growth pattern, and these are the cell generations. So you one petri plate to the second petri plate to the third petri plate, fourth petri plate, and they will have a kind of a growth speed that is attained, that will be maintained through about say 50 generations. And after that, what is going to happen that there will be a sharp decline of these cells, and they're all going to die. And this is a problem with the primary cell culture, that they are mortal. So that's the important thing, primary. So they have a finite life. There are some exceptions that we talked about earlier, that there are some diploid cell lines that were developed that can live up to 100 generations. At the very beginning of this class, we talked about that, but those are exceptions. Most of the cells will die. Now, what you can do, that without any treatment, this is going to happen. But if you plate these cells in a petri plate and you treat with a UV or a chemical mutagen, what you are going to see that these cells will immediately start dying. Instead of they are attaining a growth, they are just going to die. And why this is happening? Because you have treated them with the UV or chemical mutagen. Probably it disrupted the DNAs, broken DNAs, and most of the time this is lethal to the cells. So they're going to die. And at the very bottom of this curve, that what you can see the growth rate right here, very low growth rate. This is the y-axis is the cell growth rate. This is called a crisis period. And you may see it is not as easy that as it sounds, that at the end of this crisis period, suddenly some of the cells starts growing back. And this is what we call as the appearance of an immortal cells. And these have a very different morphology than these normal cells. And we call these as the transformed cells. And we are going to see that how viruses can induce this. Now, the most important thing that is important, that remember that to do this process, it is just not 1, 2, or 3 petri dish that you irradiate with UV or a mutagen is going to do this. You have to probably do 50, hundreds of these plate, and in some one plate, you may be lucky to get an immortal cell line. So that's one of the important things. Another thing, what happens without a treatment, it has been seen that, say, hamster or mouse embryos sometimes spontaneously get transformed in the cell culture as you passage them spontaneously. But that has not been seen in primate like in the monkeys or the human cells. And probably you can just understand that compared to the checkpoints of the cells, and all these transformation happens due to irregularities in the cell growth pattern or in the cell cycle. So you can easily understand that probably compared to the mouse or hamster, the primates, the monkeys, or the human beings have a more robust cell cycle control. That's the reason that untreated cells rarely undergo transformation. Whereas the spontaneous transformation can happen sometimes to hamster and mouse embryos. So what are these cells? These are called the transformed cells, and they become immortal. And let's take a look at what are these properties of these. They can grow indefinitely, and they require a less requirement of serum growth factors. And why this is so? Because remember that these cells actually turn on some of the genes which produce their own growth factor, and they become autocrine. So this is a cell, it is releasing its own growth factor and stimulated by that, and that's why they have less requirement of growth factors. And they also lost capacity for growth arrest upon nutrient deprivation. Say, for an example, these cells, if you just deplete the glucose from the media, it will stop growing, but they will die rather, not be able to produce ATP, but they will still try to divide. Their growth will not be inhibited by the nutrient deprivation. Grow with very high saturation densities and loss of contact inhibition. So you can see that right here. You can see that the normal cells will not grow with this. They are contact inhibited, grows on piles, but there is no contact inhibition with these transformed cells. And also different type of morphology, rounded, refractile. Anchorage dependence. They do not need the anchorage dependence like a plastic flask or a Petri plates to be attached at the bottom of these. They can grow in even semi-solid media like soft agar. They can grow on top of each other. Now, the important thing is that these immortal cells may be tumorogenic, but not always. So the thing is that the transformed cells may be tumorogenic, and then again, they could be either a benign tumor or oncogenic tumor. So how do you understand that? You take some of these transformed cell lines from, say, a mouse, you inject it to the mouse. Some of the mouse, nothing can happen. In some of the mouse, it can develop a benign tumor, or it can be developing an oncogenic tumor. But again, remember that when you're injecting these cells to a mice, the mice has to be immunocompromised. Like usually they will not have any B cells or the T cells. The reason being that if the mouse immune systems are good enough that they will immediately kill all the incoming cells. So you have to be look at the tumorigenic potential. You have to inject them to a mice which is immunocompromised. And these are some of the properties of these transformed cells. And what it has to do with the viruses, we have not seen yet. And that's going to come together. So you can see the next part is oncogenesis. And remember that oncogenesis is different than transformation. All transformed cells will not lead to oncogenesis. So let's take a look at that, what can happen in a tissue, not a cell culture, normal cells, and suddenly some of the cells become abnormal, growth is uncontrolled, and they multiply. And if this is the condition where it stays, see that this is, say, for an example, on the skin surface, this could be as simple as a skin tag, a benign tumor. But if they invade the deeper tissues or a different tissue, see that this is right here, epidermis, this is one tissue, whereas underneath, this is connective tissue. If this invade other tissues and then through the blood or the lymph go to other tissues as well, this is what we call as a malignant or invasive cancer, and the process is called metastasis. You already know that. And large number, you can easily see 8.2 million deaths per year in developed countries. And another interesting thing that you need at least 12 mutations, and that's a large number, that 12 things has to go wrong in a same cell, remember that, in a same cell, in those 23 into 246 chromosome, 12 mutations need to accumulate before a cancer can develop. And many of them can be inherited as well, and also caused by damage, carcinogen, infectious agent, and viruses. And that's where the connection is. So let's take a look at this, that the transformation, immortalization, and oncogenesis are distinct but a part of a continuum. Remember that the cells are transformed, that led to their immortalization. And then these immortalized cells either could be oncogenic or may not be oncogenic. And that's what the idea is. And what is the difference between a cell which is a transformed, immortalized, and then oncogenic or not oncogenic? This is what it is, that a cell to become oncogenic require additional genetic changes. Just a transformed immortal cell is not enough to induce cancer or oncogenic. You need more mutations to develop a cancer, say in an immunocompromised mice. So what happened, that now you know that the viruses kind of transform cells, and this provides the cell the molecular events that establish the oncogenic potential. And remember that all of these processes, no virus can do it all by themselves. There should be some mutations that is also happening in the cell to do that. So that's the overview of the transformation, immortalization, and oncogenesis. So the most important thing that you have to remember here, that the transformation oncogenesis is not required for the replication of any viruses. It is not a requirement for their transmission, for their growth, except probably one, which is shown here in a walleye dermal sarcoma virus with an unique transmission cycle. See, this walleye dermal sarcoma virus is a retrovirus, and it's an epsilon retrovirus, and it was probably the group that was discovered latest in the late 1970s. And we are going to talk about that. This is the only one where the virus propagation is related to the formation of these sarcomas, but this is the only one. But remember that for all other viruses, the transformation oncogenesis is a chance factor. It just did that process by chance. It did not have the intention to do that because it doesn't help the virus to propagate or replicate. So here we are looking at the human cancer causing an oncogenic virus. And if we just look at this, that there are two groups right here, a flaviviridae. This is a hepatitis C virus. Hepatocellular carcinoma is formed. Retroviridae, we're going to talk a lot about that. One of the example is a human T lymphotrophic virus. And you can see a good number of DNA viruses. Hepatiniviridis is hepatitis B virus, then a good number of herpes virus, polyomavirus, papillomavirus. You're going to see that most of our talk will be the retroviruses in the first part, and then herpes virus, polyomavirus, and papillomavirus. We're going to talk about the adenoviruses as well along with that. And these actually cause the cancer in the human beings. Now here, these are contributing factor for about 20% of the human virus. HIV, on the other hand, does not cause cancer by itself, but promote cancer due to the immune system failure, because it just cannot, it kills the CD4 T cells, it just cannot induce the cytotoxic T cells, and gradually is a collapse of the immune system, where some of the herpes virus that's already in there, like Kaposi's sarcoma virus, that's very common in the HIV patients, like in the AIDS patients, that can happen due to that. On the other hand, what we can see that the adenoviruses, polyomaviruses like SV40, and even pox viruses can cause cancer in other animals, but not in the human beings. These are only the human viruses. That's why you don't see adenoviruses here, because there's not. But there are some other polyomaviruses, like a Merkel cell polyomavirus, and this Merkel cell is a sensory organ which is underneath your skin. In the just below your epidermis, in the dermis, that's a Merkel cell polyomavirus that can cause these skin sarcomas. And we have no pox virus is known that can cause cancer in the human beings, but in other animals, yes. And papilloma viridi, you know that very well. There are two, 16 and 18, HPV 16 and 18 that can cause cancer. And these are mainly implicated in the cervical cancer that you know predominantly by these HPVs, and you have a vaccine for that as well. So out of these, what we're going to see that we are not going to discuss this hepatitis C virus and hepatitis B virus. The reason is that the mechanisms are not really very well known. So hepatitis C virus, I have written that in your notes section. Hepatitis C virus actually will cause persistent infection. In that case, what happens that as it is a persistent infection, it is thought that it blocks all these apoptotic signals, which can kill the cells and also increase the amount of reactive oxygen species, and that can cause a DNA damage. So reactive oxygen species is produced in the cell under the stress because it's virus infected, but the hepatitis C virus is also blocking apoptotic signals, so the cell will not get killed. So the cell sustained DNA damage, and this may lead to the oncogenesis, which is not clearly known. On the other hand, the hepatitis B virus is very interesting, that almost always you actually do see they also cause persistent infection. There is a portion of the viral DNA integrated into the DNA of the host. That is already been seen. Remember that here is one important thing. Hepatitis viridi contain a reverse transcriptase in its life cycle. It codes for a reverse transcriptase. But unlike retroviruses, it does not code an integrase, and there is not a natural way that the hepatitis B virus DNA will be integrated into the host DNA. That will happen all the time with the retrovirus because they contain the integrase, and all of these retroviral DNA upon infection will be integrated into the humans. We know that. But that is not the norm of the hepatitis B virus. This happens due to spontaneous recombination that you know that can happen in any cell over the period of time. And particularly in a persistent infection, it's a spontaneous recombination between the virus and the cell. And exactly what sequences cause this cancer is not known, but it has been implicated that a particular protein called protein X in the hepatitis B virus may be responsible for this. So we're not going to talk in detail with these, but we are going to start with these RNA viruses, mainly retroviruses, and talk about the DNA viruses, how they can cause cancer. So let's take a look at some of the very interesting story that how it all started. And I think it's fascinating. So what happened that this Peyton Rouse, that in the early 1900s, and how the story goes, that a farmer from New Jersey actually brought a chicken to him, which actually does have this solid tumor, and told Peyton Rouse, who was at the Rockefeller Institute at that time in New York, just like, can you just look at what causes these tumors in a chicken? So he started working on that, and he actually found that it is caused by this, already it has been discovered, the Rouse sarcoma virus, he discovered in 1909. And he found that this Rouse sarcoma virus is responsible for these solid tumors in 1909. At that time, nobody believed him. Nobody actually believed that a virus can cause cancer until 1966. So he has to wait 50 years before the scientific community actually acknowledged the implication of his research. So you can just well imagine that how fascinating that story is. On the other hand, there is another part of the story that you can see that this is Harold Varmus and Bishop, Michael Bishop. They identify the first oncogene, which is known as a viral SERC. It is pronounced as SERC, from sarcoma, not SRC. SERC from a sarcoma causing virus, viral SERC. In 1976. They got the 1989 Nobel. So you can well imagine, so that around this time, there are evidences that really the virus is causing these. And you had to wait until this time that when the first oncogene was captured by these two Nobel laureates. So On the other hand, there's another part of the story going on in 1950s, and this is Renato Delbecco, and you know that he discovered plaque acid. And Howard Temin was a young graduate student who actually started working in that lab. And he started working with Drosophila. He just got frustrated, and he started working with viruses at that time. So when he was working that, Temin was a dreamer, and he imagined that so in the 1950s, cell culture just being started at that time. So what he imagined creating cancer in a petri dish, and it took him about 8 years, and like a seventh year, he was succeeded to transform cells in a culture, and that changed the whole of the way that we study virology and the cell culture and the cell biology completely forever. And then he discovered, of course, the reverse transcriptase, which was another leap, which was responsible, this reverse transcriptase, along with David Baltimore. That was the key by which Varmus or Bishop could identify the gene. And we're going to talk about that. And Temin, later part as an investigator, if you just go to the University of Washington, Madison, which is a very strong virology department. There is a lakeshore along with the Mendota Lake around that UW Madison. There's a path which is called Howard Temin Lakeshore Path. You can just go around with that. So another interesting thing, this is a personal story with that I had a high school friend, and his name is Udan Guho. So he did his MD PhD from so he did his MD from India and his PhD from Albert Einstein College of Medicine from New York. And he was the one, I have only one friend probably who worked with a Nobel laureate, which is Harold Varmus. And then he was an investigator for a long time, for almost 15 years at NIH. But just recently he joined the Bristol Myers Squibb, and he is the lead oncologist. Like they are just developing oncology-based drugs. So why I just put this one, that he worked, this guy, in the late 2009, 2010, he worked with this receptor tyrosine kinase signaling in the lung cancer of the Varmus, and they just tried these transcriptome analysis that to identify some of these lung cancer genes that are responsible. So what I was just want to see that in 1976, Varmus discovered the first oncogene, and in 2006, even after 40 years, he's working in the same field trying to capture more genes. So you can see that you can just spend a lifetime and still not be done with that, what you are doing in this process. And our understanding of cancer is far more than complete at this time. So what we are going to see that how the virology now paved the path for our understanding of the cancer. And this is just the beginning. And let's see that how it actually worked. So this is the first oncogenic probe that was developed by the Varmus and Bishop by a process called subtractive hybridization. This was only possible by the discovery of the reverse transcriptase. And at that time, there was no PCR, so they couldn't do anything. So what they did, that they took this RSV genomic RNA. Remember that the RSV causes cancer, that they knew at that time, or they had a good suspect. So what they did, that they reverse transcribed. So from here, RSV genomic RNA, you can see that from a cell, you can collect the mRNAs. And from those, you can just, or the genomic RNA, and if you just reverse transcriptize, you will going to synthesize these small sections, which are usually the DNAs. So understand that what they're trying to do. They're using an mRNA probe, and then they are reverse transcribing part of this RSV genomic RNA, which are the sequence of the gene on the genomic RNA. So they're using the mRNA, they are reverse transcribing these probes, which are DNA. Remember that it's RNA to DNA. And these probes are produced. They found one RSV, and this is in the cell culture, which is a non-transforming RSV. It's a temperature defective, what they told. And what happened, that virus will grow in the culture as an RSV, but it does not have the ability to transform cells. So that's one of the things that is different from the RSV. So what they did that after producing these probes, these small DNA fragments, they hybridized these probes, all these probes with the non-transforming TD-RSV genomic DNA. After that, they passed it through a hydroxypeptide column. So it's a chromatographic. So what happened that all these large fragments, because they are now hybridized, these large fragments were in the column, whereas the unhybridized fractions, or these which are the unhybridized DNA pieces, that came out of the column. They collected this, and then they looked at the sequence and tried to find that these were used to fish out the gene, and they found that the V-circ is the gene responsible. And this was the first way. And then it was an explosion in the field that many, many genes were discovered immediately after this process by this subtractive hybridization process, which was pioneered by Varmus and Bishop. So after that, we are looking at going back to these cells that, this will give you an idea that what these transformation means and what the gene actually does to these. So if you look at that, all these cells which are transformed by the RSV, they form a focus, plural foci. So you can see the normal cell, and then you have a foci. These are the transformed cells, and this is a close-up view. You can see that right here is a fusiform cells. They are just elongated. On the other hand, you can see that this is another type. This is also, but here the foci, these cells are kind of a round and refractile right here. So you can see these round and refractile cells. So you can see that there are two different types of cells that is being transformed, both by the RSV. And why this is so? This is the difference that has been discovered later, that this is a difference in the mutation of the SERC gene. One type of mutation will cause this type of phenotype. Another type of mutation can cause this type of phenotype. So this is how they differ over the period of time. So this is also another fascinating discovery, essentially by Howard Temin. So how can a viral infection transform a cell? You can see that in this process of transforming the cell, the cytopathic effect must be reduced or eliminated. The infected cells does not die, because if the virus actually kills the cell, there is no way that you can have transformed cells. On the other hand, the viral replication must be reduced or eliminated, and the transformed cells do not produce virions, because usually when the virions actually aggress the cell, they will kill the cell and go out, and you want to stop this process. So this is one of another thing. And the cell must continue to divide, and it becomes immortal. So there is the viruses need, and so you can easily see that for the replication of the virus, the transformation is not ideal, because it is not allowing the cells to die or allowing the virus to be replicating at its full potential. But this is a phenomena which happened due to some of these viruses. And it is a chance phenomena that we're going to see time after time. So here is a walleye dermal sarcoma virus. It's a retrovirus with its unique transmission cycle. We have already seen that almost all of these viruses, we're going to talk them in detail, they are not required for the transmission. But what happens that these walleye, they are usually infected with the virus in the spring. And then there is, at that time, there is no manifestation of cancer or these dermal tumors or the sarcoma. In fall, that they are not going to grow in the fall, following the winter. In the fall, these tumors actually start developing. So, and then what happens that these tumors actually fall off in the next spring when they are going to grow and the water temperature is high at the springtime. So how this helps the virus and what it does. So think about that in the spring when the water temperature is high, the virus is actually replicating in the cell, They are not causing any cancer at that time because they are killing some of the cells. They are transmitting some other. It really doesn't cause much damage to the much damage to this fish. They just transmit. It's very high transmissibility. But over the winter, when there is not enough, the temperature of the water is less and there is not enough viruses available for the infection, they actually reduce the cell death and reduce the viral replication and form these cancers as if they are just hiding in these sarcoma, hibernating over the winter to be activated again in the replication mode in the next spring. And how it does that. So this is basically the gene of the retrovirus. You see the 2 LTR and you have a gag pulp envelope. And you have these accessory genes, A and B. So these A and B gene, when they are activated at that time, it causes these cancer, these transformation. So these genes are activated in the low water temperature, supposedly during the fall. But what happens that when the water temperature actually rises during the summer, during the spring months, these A and B is silenced by this accessory gene C, which is cytolytic, means that this will kill the cancer cells and allow the virus to replicate the gag pulp envelope to make the virus, but not form the tumors. And this is the only unique example of a retrovirus, which has something to do with the transmission of the virus is related to for cancer, but there is no other. For all other cases, the oncogenesis and transformation is a chance effect, and we're going to see that. So what we are going to see that the route for the unified theory of cell growth control, that is a present theory, is very convoluted. The first ideas are from the RSVs, that what we are going to see that 1900, the retroviruses paved the pathway. And then 1943, the cancer cell biology, they came together, and then DNA virus from the evidences all actually converged around 1960s. Before that, all of these group of scientists fought amongst each other, and they all thought that RNA virus, retroviruses are responsible for cancer. These people say that cellular genes are responsible for the cancer. These people thought the DNA viruses are responsible for cancer, and there was no cohesion among these groups, and they all converged finally in the 1960s. Essentially, once the first VSARC gene, or the oncogene, was discovered by Varmus and Bishop. And you can imagine that that paved the unified theory of cell growth in the present. So that's the importance of their discovery. So let's see that how it all started in a little bit more detail level. So here, what happened, the Rous sarcoma virus caused solid tumor, and the viral isolate from tumors cause a rapid sarcomas, but they do not cause any leukocysts or any problem of or the cancer of the leukocyte or the white blood cells. What it was found later, that most of these Rous sarcoma virus are actually defective. So, but what happened that Rous was just by chance lucky. The virus that he isolated can cause cancer. That was not a defective virus, and we will tell that what we mean by a defective virus and not defective virus. So so what happened that it was he found, and that was later found, that the isolate which was not effective contained a viral copy of a full oncogene. A viral oncogene is right there in that RSV by full. Whereas the most of the viruses which are defective only has a part of these oncogene, I'm sorry, not the part of the oncogene. You have that oncogene, but the viruses are not replication competent, because some of the part of the viruses, remember that these are the retroviruses, and if we just go back to this picture right here, so you have these oncogenes, and in the defective, either a gag pol or some of these envelope sequences, they are all disrupted. So they cannot replicate by themselves, and those are called the defective. But the Rous sarcoma virus, what he found, that there was an intact oncogene along with the full gene complement of the virus. That I mean by non-defective virus particles. And we are going to see that. And the viral genomes from the solid tumors are the recombinants that was found. Before RSVs, also another virus was in the picture. That was basically the ALV, or the avian leukosis virus. And this actually causes leukosis in the chicken, but the initial viremia caused by this develop a sporadic leukemia, about 3%. And look at this, that almost all of these non-defective viruses will always form a tumor in the chicken, whereas the ALV, almost 97% of these chicken are immune to this. They will initially get the infection, it's endemic. They will get a viremia. That means that there is a virus in their body, active growth, and they become immune. These viruses are silent for the rest of their life. But remember that these other retroviruses, they are in their genome, but they don't cause anything. But there are about 3% of these birds develop sporadic leukemia and the variety of tumors, very less. So very different. And it was found that they are most defective and all had a different host DNA into their genome, integrated into their genome. And these are the oncogenes that has been captured by these viruses. So what we are going to see, that what we mean by a non-defective virus, then we are going to see that what are these defective viruses means, and how these defective viruses can lead to cancer as well, but the chances of them being very less than a non-defective virus. That will be the next part. So here what happens, that the rapid tumor formation, say for example RSV, can happen by 2 weeks. And this, remember that these RSV is a non-defective RSV. This has an activated dominant oncogene in its genome, which is the VSERC, and the protein produced immediately when the virus replicates. On the other hand, the ALV that we have seen, that the ALV, avian leukosis virus, carries no dominant V oncogene, and ONC is for oncogene. Remember that ONC is not a particular gene. This is a generic name for any oncogene. We're going to see that there are many. What happens that a process called cis activation of the provirus turns on expression of endogenous oncogene. What it means that once these are integrated into the genome, the LTRs, which are very strong promoters, if they are integrated somewhere in the chromosome next to an endogenous proto-oncogene, they can sometimes turn on these proto-oncogene, and which becomes an oncogene. And this is by a chance factor, can take months. On the other hand, this is a HTLV, which is a human lymphotropic virus, sometimes years for this process. Carries no dominant oncogene, does not cause a cis activation of local oncogenes, and it causes a trans activation, which is even rarer in that case. And we're going to see that what we mean by the cis activation and the trans activation of these genes. So before that, before we talk about these cis and the trans activation, let's take a see that how the genomes of the transducing retroviruses look like. And again, you do not have to memorize anything, just understand that what this all means. So right here, we are looking at a progenitor, avian leukosis virus. You have a gag-Paul envelope, and of course, Rous sarcoma virus will also have the same thing. You will have a gag-Paul envelope. These are the viral sequences. Now see that this is a non-defective Rous sarcoma, which has a full length of SRC, which is the sarcoma gene inserted in there. And then there are some other sequences before and after, which is derived from that. And this just happened just by a process. We are going to see that how this is captured by the retroviruses. On the other hand, you can see that right here, you have a full length of gag-Paul envelope. So this can produce in a virus and also carry the oncogene. If you look at all of these, you can see that there is this male. These are Orel, A, or B. These are all oncogenes. But if you look at these pattern, gag-Paul envelope, all of these viruses are defective. So what is the idea that how this can happen? On the other hand, look at this as well. This is a murine leukemia virus. These are the red ones, all of them, of this RAS, ABL, MOS, RAF, these are all oncogenes. But if you look at the GAG-Paul envelope, you can see that all of them are defective. It is inserting randomly somewhere in the virus. So what is the difference between these defective virus particles and why they are even important? Remember that what happens that they are helped to replicate by a helper virus. Think about this way, that you have an integrated DNA like this, which is excised out in a cell. And at that time, it is replicating in the cell. You have another sequence in the cell somewhere else, because remember that these are integrated into the genome in many different ways. And another copy excised out itself as a provirus, which is not defective, but does not contain a cancer gene. But in the same cell, the non-defective virus particle without any of these oncogenes will produce GAG-Paul envelope, all the mRNAs that will help to replicate these viruses, package its genome, and egress through the end of the infection. And that can be transmitted to another person, and it can be integrated into that genome or a bird or whatever it is. And this is way that the Rous sarcoma virus do not need any helper virus to replicate, whereas all the defective viruses will need a helper non-defective virus to replicate, and that's how they can propagate over the period of time and also cause cancer. This is very rapid, whereas the chances of this being defective is less because you need a helper virus. That's why the chances are less in these cases. And that gives you the idea that why the defective ALV cause in only 3% tumors, whereas RSV, which is a non-defective, can cause tumors almost instant within 2 weeks. All right, so here is the process of how the oncogenes are captured by the retroviruses. See that what happened, that you all know that it is you have the GAG-Paul envelope. It is integrated into the host genome by the integrase, and here you have the LTRs and the sequence right here. Somehow, this is totally by chance. If this sequence is in some of the areas which is close to these oncogenic sequences, these mRNAs are produced from these. This is a full-length genomic mRNA produced from these GAG-Paul envelope. And at that time, sometimes what can happen that there is either a deletion or a read-through through these LTRs. So see that right here, what happens, that while the mRNA is being produced in a read-through, say from right here, it just goes GAG-Paul envelope, and then the mRNA doesn't stop. It reads through the LTR and the oncogenic sequences. And see that what happens, this is splicing basically. And now the virus actually package this oncogene. Whereas in this process, this is a deleted portion. Some portion of the viral sequence is not replicated or deleted. And this is a defective integration. And right here next to the oncogene, which is captured the same way. And then you have a virus particle, which is also defective. Now what happens that there is also in a cell, remember that these are integrated in many copies, just not one site in one place in the DNA, many copies. Some of them can have these oncogenes, defective or non-defective mRNAs produced with that. This is a plus strand mRNA. Remember that that's going to be full-length plus strand mRNA, which is the genome of the retrovirus. But some of the sequence, some of the mRNAs could be just intact viral sequence. That is also packaged with the virus particle, because remember that retroviruses are diploid. And when these viruses are infecting another cell and started replicating, at that time, what happens, that there is a copy choice mechanism. So you can see that there is always one strand is being synthesized, and it can just jump between the template, and you have a copy choice process, and by which process it can capture these oncogene. And now you have this oncogene, which doesn't have any issues with that because it is intact. It is a proto-oncogene, doesn't have the ability to cause cancer even, because it is the viral oncogene could be exactly as the cellular oncogene. But there could be additional rearrangement and point mutations can occur into this V oncogene, and that can turn it into oncogene, which can cause cancer. So this is a way that the oncogenes are captured by the retroviruses by these deletion or read through, followed by the copy choice recombination, non-homologous recombination in this process. All right, so now if you just look at that, what are these proto-oncogenes? The proto-oncogenes actually tell the cells for a go. Induce go for the cell cycle means divide. And there are more than 60 of them discovered in cells, control growth, highly recruited, and the normal cellular genes are abbreviated as cellular oncogenes. So this is your the generic name. And cellular oncogenes, cellular circ, cellular MYC, SMOS, RAS, these are the oncogenes. When the virus actually captures those and integrate them into their viral gene, and these are all retroviruses, they are called the viral oncogenes, or the VCirc, VMYC, VMOS, or the VRAS. That's what how the nomenclatures are. So let's take a look at going back to this. This will now make sense. So here we are talking about this rapid tumor formation versus intermediate and the slow kinetics, months and years. Now we know that the process of capture and how these actually happen, this will now make sense. So now remember that when a full length of these viral sequence is integrated into along with the V oncogene, viral oncogene is integrated into the genome. At that time, there will be a rapid progression of the cancer, because this is an intact virus which has a full viral sequence and the intact oncogene. On the other hand, this is what we call as a transducing virus, which is a rapid transforming virus, which can transform the cells and can cause cancer. Whereas the non-transducing viruses are slow, intermediate, which is months that we have seen, or even slower. So how this actually happened? This happened in an entirely different mechanism. In this process, you can see that you have an entire viral sequence integrated close to a cellular oncogene. These are not capturing the oncogene at all, like the transducing viruses, but they are close to some of these oncogenes, and these strong promoters like the LTRs may express these oncogenes more than a level than normal, which can sometimes lead to the formation of tumors. On the other hand, here you can see that sometimes what happens that right here, there is also an oncogene that can be a part of these the retroviral sequence, and sometimes the activation of this is due to a trans activation. And you can see that this is a cellular gene, and this trans activation is a very long distance activation of cellular genes, for example, interleukin-2 or interleukin-2 receptors. So these LTRs are very strong promoters. Remember that, very strong promoters, and they can actually entrance maybe thousands of base pair away, and by DNA bending, they can activate some of the cellular genes. And these are the slowest out of all. So here you can see that in this case, the oncogene is directly under the LTR, and it will express every time the virus is actually produced, whereas these are the cis or the trans activating, and these are seen in the non-transducing retroviruses as such. And remember that it is a viral mistake or byproduct. The virus didn't intend to do that. This transactivation virus didn't intend to do that, and these are just a byproduct of their process, except the Wallei dermal virus. So here is also another mechanism. The same thing. Here is the different insertional activation of the c-MYC by the ALV, the avian leukosis virus, and all these great things shown that where the insertions actually can take place. And how this insertion can happen? There are two ways. One is a promoter can be inserted. You can see that right here is a cellular oncogene, and a promoter can be inverted. And on the other hand, an enhancer can be can be put into that before a cellular oncogene. And either it could be in cis or it could be in trans. This LTR is activating in trans this, whereas here this LTR is activating or promoting the transcription of these C oncogene. So one is an enhancer insertion can do that. Another is a promoter insertion can do that. And so how these promoter or enhancer insertions actually do that? Remember that we have already said that these sequences are just going to be integrated into the genome very randomly. That's number one. And another very important thing that we have talked, that we have the retrotransposons in our genome, which allow portion of our genomes to move around in our cells. And some of these movement by these retrotransposon can cause this insertion of these enhancers or promoter upstream, downstream of proto-oncogene, which can turn them on by this transcription, and but it is a totally chance factor. All right. So after that, what we are looking at, that how the DNA viruses are coming into the picture after this process. So the DNA tumor virus, the first oncogenic virus was discovered was a papillomavirus that causes the warts in the cottontail rabbit. And this was discovered in 1933 by Richard Shope. And also then in 1953, polyomaviruses, pox viruses, these were all implicated in cancers or DNA tumor viruses. And this is what we are seeing, that this is actually, you are seeing that some of the warts are growing from the head and the nose area of these rabbits. And this is probably known as, or this is known as a jackalope. This is a mythical creature that you can just look it up. And this jackalope is probably the imagination from this type of papillomavirus infected rabbits itself. And that doesn't kill the virus, it just falls off. This is like skin tags, but just like antlers or projections of from their mouth and nose areas. But what happens that it has been seen that all the DNA tumor viruses have a very rare transformation and tumorigenic potential compared to the retroviruses. And why it is so, that we are going to see that. So the adenovirus is a family of transforming DNA virus, many human serotype, but this is the most important thing, do not cause cancer in humans. About 18 tumors in hamster, 7 to 11 poorly tumorigenic in hamsters, and the tumors transformation cells like a polyomavirus and papillomavirus, and they're extremely rare events. So that's the take home, that they do not cause cancer in the humans and they are rare. And we will just see that what is the implication of this. On the other hand, what it was found that these transformed cells by the polyomaviruses, papillomaviruses, adenoviruses, they all contain these tumor antigen. Now, we talked about so much of the large T antigens as before the LT, you're probably tired of it. Now you know that why it is called LT. T comes from a tumor antigen. Why? Because if you find a tumor which is caused by a polyomavirus, if you just take that tumor and find a viral gene in its DNA, it's going to find an LT in there. So this large LT, small t, these are the generic name. Remember that tumor antigens is a generic name. It could be large or small. Similar T antigens in the papillomavirus, particularly 16, 18, are known as E5, E6, particularly the E7 genes. In adenovirus, the T antigens are the E1A, namely. All of these viral genes are all different proteins. Now, remember that main conceptual difference, the retroviruses captured a proto-oncogene, but these are all different proteins. These are not proto-oncogenes, but these are shown to be present in the tumors. And what is going on in here, that's what we're going to see, that how these proteins are basically inducing the tumors. So unconnected discoveries, but it was critical of understanding the link between the virus's transformation of the cell cycle, particularly the DNA viruses. So the SV40T antigen found in the tumors. Transcription of a set of adenovirus early gene, and we have seen this, the E2 gene is basically required for the E2F transcription. We have seen that before. E2F is responsible for the transcription, and a family of proteins called the E2F family. And E2F is found to bind to a cellular protein called a retinoblastoma protein. And we are going to see that it was discovered, this P53, RB, E2F, all are critical players in the normal cell cycle. And we're going to see that what role they play and what role they have in the tumor formation by the DNA viruses. These are played by the DNA tumor viruses, but not by the retroviruses. So the first thing is that just a quick overview of the cell cycle, that what happened, that a cell must get a mitogenic signal. And this could be through a growth factor or a receptor tyrosine kinase signaling, something like that. Once this mitogenic signal goes on, it tells the cell to go. And at that time, the cell actually doesn't allow immediately to go. It holds the cell in a restriction point and checks the environment. And once it sees that all the environment, all the nutrients are okay, the DNA is not damaged, everything is good, at that time, it will release the cell from the checkpoint so that the DNA synthesis can happen in the synthetic phase. It will go to G2 and go into the mitotic phase. So to hold the cell at the restriction point, this retinoblastoma protein, RB, is responsible. But what happens that, as you have seen, that if both copies of this RB gene are lost, it develops retinal tumors. Particularly the cells these are usually in young children, this happens. These cells are gone by the age of 5. But this is a recessive oncogene. That means that both the copies, if one copy is defective, it is good enough that one good copy can produce this RB and can hold the cell cycle at this restriction point. But if both of the alleles of the two copies of your chromosomes are gone, then that can develop tumors in young children. So this is very important. RB is, so take home message from that RB is very important to keep the cell cycle in control by holding it at the restriction point. So here what we are looking at, that now how you pass through the restriction point in the mammalian cell. So to pass through the restriction point, there is something that you have to do. First, you have to get a signal. And here you can see that you do not have to go through the detail of that. This may be, if you have gone through the cell biology, you probably know that this is receptor protein tyrosine kinase, and you are getting a signal, which is a mitogenic signal. This will be transduced through this, the GRB, to the RAS, and through the MAP kinase. These are all transcriptional activator. Just as you know that this is basically a signal transduction, which will activate the transcription of this, that is good enough. Signal transduction, individual names are not required. So what has happened due to this transcriptional activation, the cyclin D1 and cyclin-dependent kinase 4/6, that is not 416. Cyclin D1 and cyclin-dependent kinase 4 or 6, these are basically produced. Their transcription goes up. What happens that you are also going to see that these RBs are going to keep in check the G1 to S phase transition in a phosphorylated form. And we are going to see that how this phosphorylated form is made unphosphorylated or hypophosphorylated, less phosphorylated, for the cells to enter into the cell cycle. So this is just only gives you an idea that these are necessary for the G1 to S phase transition. Now let's take a look at what is happening here. So here we are looking at some cells right here where it is not undergoing cell division. It's held at the restriction point. So what is happening at this point? You can easily see that at this point, the E2F is bound to the dimerization partner, DP. You have seen that beforehand. And RB is bound to them. In this condition, right here, the HDAC, histone deacetylase, is recruited, and this will keep the DNA in a tightened form, and there will be no DNA transcription. So now what is happening, that gradually this cyclin D and 4,6 are not going to form. But even at that time, there is a check from another protein, which is called INK4. But what cyclin D and CDK4/6 will do, because this is, remember that the CDK is a cyclin-dependent kinase, this will phosphorylate in the cell. Remember that this has nothing to do with the virus yet. In a cell, it will phosphorylate the RB, and now once the RB is phosphorylated, it will fall off and take the HDAC away, and you will have DNA synthesis. And then cyclin-dependent kinase is also feedback into the loop. So the transcription will also produce the CDK cyclin E, cyclin A, more E2Fs, and that feeds into the loop. So basically what happens, that cyclin D4/6 transcription, due to the mitogen signaling, will phosphorylate the RB to inactivate it. And that will go into the DNA synthesis. Also, another thing that what can happen, that you can see that the cyclin A also activates cyclin A CDK2, NFS promoting complex, increased cyclin, and mitosis. So this is even downstream. So all you have to do from here, that phosphorylation of RB is basically responsible for growing out of the restriction point. Now we are out of the restriction point to the S and the G and the mitotic phase that we have just seen before. That is the important thing. So what is happening, that the T antigens, the tumor antigens, whether it's SV40 or adenoviruses or other, they kick these quiescent cells, which are on hold at this restriction point, into the S phase. And also we're going to see that they activate another regulator, which is called the P53. And we're going to see that how this happens. So here, this is the same picture, but what happens, see that right here, this is E1A, is the adenovirus tumor antigen. LT, SV40 LT, this is the SV40 tumor antigen. E7, this is the tumor antigen of human papillomavirus. What it does, it binds to the RB the same way the phosphorylation take away the RB, the same way this tumor antigen binds to the RB and release the HDAC from the E2FDP complex. So it kicks the cell into the S phase transition. So this is the same process by which all the tumor antigen helps the cell or the quiescent cell to move into the DNA synthesis and the mitosis. This is required for the cell because many of the viruses, as you know, don't replicate in the cell. And that's the reason that they do this. So here, on the other hand, the entry into the S phase decision is more control. And this is by a P53, which is known as the guardian of the genome. So what it does in a normal cell, let's take a look at this left column. In a normal cell, this P53 is produced, and it is bound to this a protein called MDM, and rapidly it is ubiquitinated, and by this ubiquitination, it is directed to the proteasome and degraded. It is degraded with less than 30 minutes. So P53 is very unstable in a normal cell. But what happened when they sense a DNA strand, double-stranded DNA break, it activates a protein called ATM and then a CHEK2, or a checkpoint kinase 2, which phosphorylates the P53. And now it cannot be degraded by the proteasome, and it recruits a P300CBP, CAMP binding protein, into a tetrameric complex and a transcription site. This is basically recruitment of a transcription complex, and this will produce responsive promoters, which are going to produce this P21 CYP, BAX, MDM2, which will cause cell cycle arrest. If it cannot arrest the cell cycle, it will lead to apoptosis or killing the cell, which is BAX is one of them. So basically, if there is a DNA strand break, This is how the cell keeps the cell cycle in control. It's not going to replicate the damaged DNA. So the viruses must counter the p53, and we are going to see that how the viruses counter these p53. So here you can see that, again, you do not need to memorize each one of them, but here at the bottom, we are seeing that in a normal condition, what is happening? p53, when they are accumulated in the cell during the DNA damage in a cell, it will either lead to apoptosis, cell cycle arrest, and you can see the apoptosis. And these are done by the repression of transcription binding protein, increased p21 CYP that would cause the cell cycle arrest, and BAX-PAF1. These are some of the protein which also are pro-apoptotic protein. So what on the adenovirus, papillomavirus, or the polyomavirus, these are the tumor-causing DNA viruses. See that what is happening? p53 is now bound to these E4ORF6, and this causes all these proteins will. So this is your E4ERF6. This complex will recruit these ubiquitin proteins right here due to this, and that will cause the proteasomal degradation. On the other hand, you can see right here the HPV16 or HPV18 E6 proteins that would also cause the ubiquitination of the p53 and degradation by protein by proteasome. On the other hand, the SV40LT and the adenovirus E1B does it a little bit different way. It just binds to this tetrameric p53. This is called the sequestering. And by this process, it will not allow this stabilized p53 tetrameric to bind to the DNA. If it doesn't allow to bind to the DNA, it will block the transcription of this. Because remember that we have seen that the tetrameric stabilized p53 is responsible to recruit the transcription machinery to produce these apoptotic gene or the cell cycle arrest gene, and that will produce those proteins necessary for them. And the sequestering means that they are just blocking the binding of these proteins to the DNA and blocking this process. So one process is ubiquitination and proteasomal degradation, and another process is basically sequestering. So two more mysteries remain: why all the viral genes except T antigen deleted and turned off in SV40 polyoma adenovirus transform cells, and why it is so rare. So here is the thing, that what is the important thing: the lethal late gene must not be expressed in the cells that needed to be transformed, because this lethal gene will kill the cell, and there is no meaning for that because the cell will not be transformed or not form tumor. It has to keep the cell alive. So this is only possible by a rare spontaneous deletion of the late gene in the viral genome. On the other hand, the T antigen of these viruses must be constitutively and transmitted to every cell so that the cell cycle can counter the RB and bound to the RB for the replication, and also counter the p53. So both of this event, random mutation and T antigen recombination to the DNA, must happen, and that these are two events, very low probability events, and that's why it is so rare. And these events are not required for the normal viral life cycle of the transmission, and they were discovered by infecting the wrong host. And see that why they were found in the wrong host. Think about that. A human adenovirus infecting a hamster or a mouse, the cell condition is not ideal for these viruses to replicate in those cells. So what the virus will try to make a condition in that cell so that the cells actually divide. In that process, it may kick those cells into a tumorigenic form in that process, because it is not an ideal condition for the virus to replicate. Whereas in a normal cell, the virus is absolutely happy in replicating, killing the cells, going to another cells, again killing the cells. That is not happening in a different or a wrong host. So chances of the tumor formation in a wrong host is more. But now we understand that even in the natural host, these rare events may occur, leading to tumor genesis, as in the human papilloma viruses. So again, remember that DNA tumor viruses, this is just a recap, must start the cell synthetic machinery to make the DNA. The T antigens turn on the cell cycle because the G1S phase through inactivation of the normal inhibitor RB, and inactivation of P53 blocks the apoptosis. This is all of these need to happen. The reason for that, if lytic events are blocked, the cells making T antigens continue to divide and they are transformed. And they are on their way to become the cancer cells, a secondary effect. So finally, the final thought, I will just take a second. The transforming retroviruses actually turn on the dominant oncogenes. So say, for example, MYC, RAS, SERC, these are the genes. When these are turned on, they are going to cause cancer because these are the dominant oncogene. They are off all the time. They are only on when they are needed for the cell to replicate. But retroviruses turn them on, so it's the cell cycle goes on the go. On the other hand, DNA tumor viruses actually activate the recessive oncogenes like the RB. These are DNA viruses turn on constitutively on tumor suppressor gene. So RB is a tumor suppressor gene. It is constantly on and checking the cell cycle at the restriction point. When the virus comes in, the DNA viruses, it will sequester or turn off the RB by binding to the T antigens, and that will lead to the cell cycle. And this is how the difference between how the proto-oncogenes are activated by the retroviruses, whereas the tumor suppressor genes are turned off in the DNA viruses. And this event is much more rare compared to the activation by the proto-oncogenes by the retroviruses.